Hi, my name's Kathy LaFay, and I'm a research biologist for NOAA's Northwest Fisheries Science Center. And I'm leading a large-scale project with many collaborators throughout Alaska and beyond um, to study harmful algal blooms in Alaskan waters. And the goal of this project is to study these toxic algae species that are located in Arctic and subarctic waters and determine how those toxins that are produced by these algae move through the food web and affect marine resources like fish, marine mammals, and other wildlife. So this is a large scale project, as I said, and we are taking samples from cruises, research cruises during the summer and getting comprehensive sample sets of all layers of the food web. What we'd like to do with you is engage local communities to help take additional samples so we can get at the seasonality of these blooms. So the next section of this video will show you how to take these weekly samples that we can then use to count cells to see when they're present and when they're absent over the seasons or over the entire year. So thank you.
First off, you're going to use your bucket and rope to collect a water sample from wherever your site may be it dock, boat, or shoreline. Before anything else, you're going to record the seawater temperature with the hobo logger in your field kit. Place the logger in your bucket for at least 3 minutes and make note of the time. While you are waiting, record the site information, date, time, and sample collector on your datasheet. The next step is to rinse out your sampling container with a little bit of extra seawater from your bucket. This is simply to remove any debris from a previous collection. After rinsing your container, you can then proceed to fill it up to the 2-liter mark. Once you've fully collected this sample, you have two options. Either you can process this right in the field where you are, if you're in a good spot, or you can process this elsewhere. When transporting your 2-liter sample, be sure to keep it secured in your cooler with ice. Once you are in a good spot to start processing this sample, the first thing you want to do is collect filtered wash water in your squirt bottle. To do this, you will pour a small amount of your 2-liter water sample through the 15-micron mesh sieve and save it in the squirt bottle. The mesh of the sieve will capture any cells that are larger than 15 microns, and this includes the harmful algal species that we are interested in. Any water that flows through the sieve is clean of these cells and can be used for rinsing. When your squirt bottle is full, you can pour the rest of that 2-liter sample through the sieve. While indoors, it's best to do this over a tub or sink. If there is a lot of debris or plankton in your sample, it may take a minute for this water to flow through the sieve. To help speed up this process, you can tap the side of the sieve using your wash water container, or even tap the bottom with your finger to help force the rest of the water through. Once you have all the water through your sieve, there should be cells stuck on the mesh screen inside, and you want to condense those all down to one side by holding the sieve at an angle and squirting that mesh. Using your funnel, you're going to transfer those cells from the sieve into your 15 ml tube. You want to hold the sieve at an angle and use small squirts of wash water to dislodge everything on the mesh screen and wash it into the tube. After this transfer, slowly and carefully fill the tube to the 14 milliliter mark. If at any point the tube overflows, you should take a new 2-liter sample and start over. But if that is not possible, continue processing the sample and note down the spillage on the log sheet. You are then going to add 6 drops of your Algal solution to the tube, and this will preserve any plankton in the sample for future analysis. Algal is semi-toxic and can stain your clothes, so try not to get it on you. After the drops have been added, secure the cap and invert the sample at least 7 times to thoroughly mix it. You are then ready to write up 2 labels for your sample. These labels should include the date, the site, and your initials. One of them will go on the tube directly. After it is labeled, You're then going to use some parafilm to secure the cap and prevent any sample loss. I do this by ripping off a small corner so I can remove the back and then holding this stretchy stuff to one part of the lid and pulling it around the sample. You want to be sure you are going with the twist so that you're not dislodging the lid in the process. This ensures that no sample is lost. Then you're going to cover it with aluminum foil, and this is to prevent the sample from degrading.
Burrito, tube.
You are then going to place another label on the outside. Now this sample is ready to be stored, which you can do so in a regular 4°C refrigerator until you have a handful to ship to us. Once your samples arrive in the Anderson lab, they are processed with a fluorescent probe so we can identify and count the harmful algal cells under a microscope. Knowing the number of cells that were in your 2-liter water sample allows us to estimate the overall abundance of these harmful algae in the water at your sites.
Again, the goals of this research are to study harmful algal blooms in Arctic and subarctic waters, answer the questions of where are these blooms, how toxic are they, how much toxin do they produce, and how do those toxins move through the food web. The two main groups of algae that we're interested in are Alexandrium, that's one group that produces saxitoxin, as well as some other toxins that are responsible for the neurotoxic illness in humans known as paralytic shellfish poisoning. The other group of algae that we're studying are Pseudonychia. They produce domoic acid, which is known to cause a neurotoxic illness in humans known as amnesic shellfish poisoning. And we know that these cells are present in Arctic and subarctic waters and that their concentrations are related to environmental conditions. So when do we get bigger blooms? When do we get more toxins? Those are the things we want to investigate. And we're looking at that in the context of climate change. So warming oceans and loss of sea ice.
Thank you again for watching this video and participating in the project. It would not be possible without you. Please contact us with any questions you may have using the contact sheet in your clipboard.